Okay, well, I'd like to welcome you all to this year's celebration of global fungi. This is the fourth of our global webinar series to celebrate fungi. And of course, we all know that fungi are crucial to the functioning of all of Earth's ecosystems and indeed to our everyday lives as humans. And today, as in the previous 3 years, we have talks from east to west of the planet, though not from Antarctica this year. We've got 3 sessions during the day. Our first session, i.e. the present session, is with speakers from Asia and Australasia. Our middle of the day session is from Africa and Europe. And then the third session of the day is from the Americas. If you haven't been able to watch all of these talks, then they are largely being recorded, and most of them will be available at a later date on the BMS website. Now, each of the sessions has 4 speakers, and the 4 speakers have about half an hour each to talk, and there won't be any questions after each individual talk. All of the questions will be reserved to the end. But if you do have questions you would like to ask, please put them in the chat as you go along, and I'll try and note these down so that I can read out the questions in 2 hours' time. And please do ask whatever you feel you need to ask. And I will also ask the speakers if they can answer each other's questions and to be involved in the discussion. So we have a very nice discussion at the end of these 4 talks. I'm sure you're looking forward to these talks as much as I am. And so on that note, I will stop introducing the general session and get onto the nitty-gritty of the day and introduce our first speaker. So our first speaker is Yuki Ogura-Tsujita, and I've pronounced that incorrectly. I'm sure, Yuki, please forgive me. We welcome you very much. Yuki is an associate professor in the Department of Agriculture at Saga University in Japan. And Yuki researches into mycorrhizal symbiosis of orchids and mycoheterotrophy. So I'd like to hand over any moment now to Yuki to tell us about the giant mycoheterotrophic orchid, Erythrorchis altissima, and to tell us about its unique association with wood decay fungi. So welcome everybody, and in particular, welcome Yuki, and over to you now if you'd like to share your screen.
Okay, thank you very much for introducing me. I will share the slide now. Okay, and then, okay. So, okay, my name is Yuki Ogura-Tsujita. I'm now talking from Japan, and Japan is now 5 o'clock p.m. And I'm our associate professor in Saga University. So the Saga city is a quite minor city. So I showed on the map. So I was located in the Saga city here, and it's quite minor city even for Japanese. So maybe if you say I went to Saga, but everyone will ask you where it is. But we have the International Balloon Fest next month, and there are many historical places in Saga City, so it's very welcome to visit our city. So I studied the mycorrhizal association, especially the orchid and mycoheterotrophic plants. So firstly, I want to introduce about the mycorrhizal symbiosis. So this is a symbiotic association between fungi and plants. So fungal hyphae attach the plant roots and form quite unique structures with fungal hyphae and plant root cell. So we call it mycorrhiza, and associated fungi are called mycorrhizal fungi. And surprisingly, more than 80% of land plants have a mycorrhizal symbiosis. And the plants synthesize sugar, the organic carbon, through photosynthesis, and plants use sugar for their growth. But the plant also use the organic carbon, like sugar, for mycorrhizal symbiosis too. And plant give the organic carbon to fungi. And fungi give the plants like mineral, like nitrogen and phosphorus, and waters. So in most cases, mycorrhizal symbiosis is mutualism. It's a give and take. And yes, we can say they have good relationships. And maybe you have experience like this especially in your childhood. You have a nice friend and have a good relationship, but sometimes fight each other and it become not good relationships. So this kind of thing sometimes happen, the mycorrhizal symbiosis. This figure show the good relationship, but this one is not good relationships. So did you find the differences? Yes, the direction of the carbon flow is reversed, and the organic carbon supplies from fungi to plants. And you can see the both arrow, the direction is from fungi to plants. So it's kind of the parasitism. It's a parasitic plants and fungi. So please imagine that what's happened when plants parasitize the fungi. So the answer is plants stop photosynthesis because they don't need photosynthesis because the fungi give the all carbon to plants. So it's not necessary anymore. And what's happened when plants stop photosynthesis? So the plant lost chlorophyll. So chlorophyll is a green pigment that plants have for photosynthesis. But photosynthesis is, they stop photosynthesis. So the chlorophyll is not necessary. So they lost it. And next, they lost leaves. So plants normally extend the leaves and they need sunlight for photosynthesis. But plants stop the photosynthesis, they don't need the leaf anymore, so they lost it. So how will they look? It's not green, no leaves, So this is the answer. Yes, these photos show the parasitic plants on fungi. So it's quite unique and strange shape. These plants are living with subterranean organ like a root and the rhizome underground. And when they mature, they appear above ground and flower. They are not green and no leaves, so quite strange. And we call such kind of plant as the mycoheterotrophic plants. And nearly 500 species exist in the world. So most mycoheterotrophic plants are very tiny size. So this photo shows the kind of mycoheterotrophic plants named Sciaphila nana. This Japanese coin show the 2 centimeter height. And so you can see that this plant height nearly 4 or 5 centimeter. And some MHP become large, like about 20 or 30 centimeter. So today's my topic is the world's largest mycoheterotrophic plants, Erythrorchis altissima. So this is an orchid and the stem climbing on a tree. And surprisingly, this plant stem lengths become 2 to sometimes 10 meters, so quite huge MHPs. And this species distribute in tropical and subtropical Asia, but are quite rare plants in the forest. So this is E. altissima. The stems are climbing fallen trees. And this is a photo of the flower. And we can understand this species of orchid, quite yellow and beautiful flower they have. And this photo shows the mycorrhizal root of E. altissima. It looks like kind of other organisms, not plants. But when the E. altissima root start mycorrhizal symbiosis, the root branched and become very thick and become this kind of structures, and the color turned red. So firstly, we explore its mycorrhizal fungi. So we identify their mycorrhizal fungi molecularly by using the ITS region sequences. And we collected the mycorrhizal root from 3 different islands located in Japan. And we collected the total 6 populations and 20 individuals and identify the mycorrhizal fungi. And this is a habitat, and there are no trails, so we're walking around the forest floor and try to find the E. altissima. And we found that 37 fungal species from the mycorrhizal root. Mostly are wood-decaying fungi, and it's including in 9 fungal orders. So we found that the E. altissima is associated with a wide variety of wood-decaying fungi. So we published this data on the 2018. So in the detailed data, please check our paper. So this data are the 2018, but we still continue the identification of E. altissima mycorrhizal fungi. And this is a commonly found fungi. And the Keratobasidium and Torrichaptum and Fiscopolia and Flavia. Now we find the Flavia is the most common fungi from mycorrhizal root. And Seripolia and Schitinostroma and Neonosopanus. And some fungal sequencing are interestingly, the sequence completely matched to Bioilluminous fungi Neonosopanus nambi. And sometimes we luckily found the fungal fruit body just beside the mycorrhizal root. So we took this photo last year's survey. We found the yellow fungal fruit body on the just next to mycorrhizal root, and the sequence from fruit body and root are completely matched. So we can identify the species name. It was the Flavia chrysocleus. So sometimes we luckily identify the species name too. So now we found the E. altissima is associated with a variety of wood-decay fungi, but they are the generalists, wood-decay generalists. But the generalist MHP is quite and very rare. So most mycoheterotrophic plants prefer a very specific fungi only. So this generalist type MHP is a very interesting findings. So this is also our findings. So this is a Europhia zoringeri. It's a mycoheterotrophic orchid in Asia. And this plant exclusively associate with Subchilera candriana. We collected mycorrhizal root from 7 sites from 3 countries and checked the 12 individual, but all the Subchilera candriana. So it's interesting, they exclusively associate with a single fungal species. So very high mycorrhizal specificity. So, the mycoheterotrophic plants with generalist type is a very new findings. So some Japanese, so our research topic become the news topic at that time and quite open in our website. And news title was Climbing Orchid, Eat Mushroom and Grows Huge. It's quite attractive news title, and the quite attractive for many people. But I check the public comment about in this news and find that the climbing orchid eats mushroom and grew huge. It sounds like some famous Japanese TV video games. And most comments are about the video games. Well, I was very interesting and laughing, but yeah, but I'm happy that everyone can know about this kind of things. And we also check the identify the carbon source of E. altissima. This is the result of stable isotope signatures of E. altissima. And today our time is limited, so I skip the detailed explanation, but we've identified the E. altissima's carbon source is from wood-decay fungi. Yeah. And also, we also find that E. altissima is associated with wood-decay fungi, but some mycoheterotrophic plants named Gastrodia confusa, they gain the carbon from leaf litter decaying fungi, Mycena species. So this is also the isotopic signatures, and this figure shows that Gastrodia confusa gain the carbon from litter decaying fungi. So before publishing these data, the mycoheterotrophic plants are known as the associate with arbuscular or ectomycorrhizal fungi, but we found that some mycoheterotrophic plants also associate with leaf litter or wood-decay fungi. They are the important carbon source for mycoheterotrophic plants. So this figure shows that the forest carbon pool, so 42% are living biomass, like a tree or plants, and 5% are leaf litter, and 8% are dead wood. And mycoheterotrophic plants associated with arbuscular or ectomycorrhizal fungi, the carbon source is our living plant biomass. So because ectomycorrhizal or arbuscular mycorrhizal fungi, they are mutualistic association with photosynthetic plants. So their carbon source is a living plant. So, the carbon flow is of the living photosynthesis plant to ectomycorrhizal and arbuscular mycorrhizal fungi to mycoheterotrophic plants. But in case of E. altissima, the carbon source was quite different. So they gained the carbon from leaf litter or wood decay, decaying wood. So the forest carbon cycle are quite different between these two types. And my next topic move on this the leaf litter and dead wood material are leaf decaying fungi and wood decaying fungi associate mycoheterotrophic plants. So mycoheterotrophic plants can only survive where mycorrhizal fungi exist because they totally depend their organic carbon on the mycorrhizal fungi. So the plant life history and plant ecological characteristics will be closely linked to the fungal characteristic of mycorrhizal fungi. So our hypothesis is like that, the mycoheterotrophic plant with wood-decay fungi. So the carbon source from a single decaying wood is limited. The wood become totally decayed and there are no carbon source anymore. So mycoheterotrophic plants may disappear from their habitat due to nutrient depletion after their host, that wood, has completely decayed. And also the mycoheterotrophic plant with wood decay fungi need to complete their life cycle before disappearing and need to move away from that place and to new deadwood. So they need to colonize neighboring log via spread seed. And for the mycoheterotrophic plants with little decaying fungi, these plants not necessary to move away because as long as the tree lives, the leaves will fall in the same place. So they can stay in the same place for long years. So we compared the life history and ecological characteristics between these two plants, the mycoheterotrophic plant with wood decay fungi and with little decaying fungi. So this is the E. altissima. So we went to the forest every year and find the E. altissima plant and tagged plant ID number. So right arrow shows the climbing stem of E. altissima, and left side is a tag for plant ID. And we visited these plants every year and check whether it survive or not. And this, so we, there are 3 cohorts. And first one, we check the 34 individual from 2015 to 19. And next cohort, we check the 17 individual from 2016 to 19. And the next 25 individual from 2017 to 19. And this figure show that each color show the survival period of the above ground stem after tagging. And we found that nearly 70% plants died within 2 years. So they are very huge, but not live so long years. And we visited the same place many years, but we couldn't find the new individual from the same place. So maybe they moved away in that place. And in total, we checked the 76 individual, and we found that the 47 individuals are flowering, and most of them make fruits and maybe spread the seed. But among 47, 33 individuals are died next year we visited. So most of flowering plants died within 1 year after flowering. So I think it's because of the nutrient depletion of host tree. And our results support the hypothesis. So this is a Gastrodia confusa research. So these plants associate with or live little decaying fungi in mycena, and they inhabit the litter-rich floor of bamboo forest. And we set up the study plot, 12 and 45 square meter, and mapped the distribution of fruiting plant for 6 years. So this is a map, and fruiting plants appeared every year from these 6 years and in the permanent plot during all monitored years. But number of fruiting stock are varied from 18 to 108, but we can find this plant every year. And surprisingly, each individual appears in the exact same place every year. They don't move away like a altissima and stay for at least 6 years. And we also find that the Gastrodia confusa, they clonally propagate from tubers. So one mother tuber produce 2 or 3 tubers, daughter tuber every year. So they become double or triple every year. So I think they fruit very much, so they might be propagate both seed and clonal propagules. So all our data support the first our hypothesis. So the Gastrodia confusa associate with litter decaying fungi or stay same place for a long year because the carbon source is a leaf from bamboo, and continuous leaf fall are expected every year. So maybe its leaf decaying, litter decaying fungi also stay the same place for a long year. So these plants can stay same place. And in the case of altissima associated with wood-decay fungi, they moved away and they immediately matured and spread the seed and moved to new log because before completely decayed. So, and also the clonal propagation. So if altissima, if the mycoheterotrophic plants with wood-decay fungi make clonal propagules, I think it's not meaning because the carbon source are quite limited in a single decade wood. So if they make clonal propagules, the propagules cannot survive for long years. But for the MHP with litter decaying fungi, so clonal propagation had a nice meaning because they can stay in the same place for long years. So it's quite effective for their propagation. Yeah, and we publish this data in this paper. So if you're interested in, please check these data. So in the last, I would say thank you for everyone who support of these studies, especially the Professor Gerhard Gebauer in University of Bayreuth for isotope signatures, and Mr. Tetsuka, who support the field work. And I also said thank you my student who working on the altissima work, the Horikawa and Kubota and Anand, and also the other people who support this project. Thank you very much for listening. Okay, I finished my presentation, so move to Lynne.
Thank you very much, Yuki. What a lovely start to the day. I think that was absolutely fascinating. And nobody's put any questions in the chat yet. However, I have got loads of questions. Please, other people put questions in as well. I mean, that was so beautifully presented, fascinating topic and very clearly presented. Thank you so much. So we'll discuss all this in about an hour and a half's time. So now we can move on to our next speaker. So thank you, Yuki. Our next speaker is Elle Bowd. Elle is an early career research fellow at the Australian National University in Canberra. And she's interested in biodiversity responses to fire and anthropogenic disturbances and restoration as well. And earlier this year, I had the pleasure of co-authoring a book chapter with Elle on the importance of large and old trees for fungi. And Elle has very kindly agreed to talk on this topic to us now. So welcome, Elle. And if you'd like to share your screen now.
Thank you very much, Lynn. I'll just share my screen. Okay, so as Lynn just kindly introduced me, my name's Elle and I'm a research fellow and ecologist at the Australian National University here in Canberra at 7 p.m. now. And I'm very delighted to talk to you today about the book chapter that I've been working on with Lynn and others, the importance of ancient trees for fungi and fungi for ancient trees. So I'm going to start today by giving you a bit of a background. We know that fungi form these megadiverse and abundant communities across all terrestrial ecosystems, even Antarctica. Fungi make up a major proportion of global biomass, more than 12 gigatons of carbon. And just to put this in perspective, this is more than 6 times that of all of the animal global biomass. And of course, a lot of the fungal biomass is underground. In just a handful of forest soil, we can find evidence of thousands of fungal species and more than 100 kilometers of fungal hyphae. It's estimated that there's between 2 to 3 million species of fungi, but only 10% of these have been formally described. And that's because fungi are pretty cryptic. They're quite difficult to study because they're often well hidden in whatever they're living in, feeding on, or growing in. So we've really only just begun to scratch the surface of the ecology and ecosystem function of many fungal communities. But recent advances in molecular methods, such as DNA, have helped us pave the way for the exploration of hidden fungal communities, including those on trees. Now, fungi form connections with trees that vary across space and time and with individual tree species. These include relationships where the fungus can be free living independent of the tree, in the soil or on the soil surface in the leaf litter. Fungi can also form epiphytic relationships with trees where they occur on tree roots, in leaves, or on the bark of the tree. Fungi can also be endophytic, and these are some of the species that we don't really know a lot about. They occur in the living plant cells of tree tissues, bark, roots, and shoots. Some of the relationships that fungi form with trees include those that are symbiotic. Mutual resource exchanges, such as mycorrhizal fungi, which form relationships with up to 90% of plant species. Another type of important relationship between fungi and trees are saprotrophic relationships. Saprotrophic fungi can break down and recycle dead organic material, which releases nutrients that living trees and other microbes can uptake and utilize. Another type of important relationship is a pathogenic relationship where the fungus can extract nutrients, resulting in the death or reduced fitness of a whole tree, or perhaps specific tissues of a living plant, as well as animals and, of course, even other fungi. Now, fungi can form relationships with trees of any age, but those that they form with ancient old trees are really very unique and important. But you might ask, what makes an ancient tree special? And that is, it has ancient features. These are really distinct features of an ancient tree that can take centuries to develop and can be lost in just seconds. And we'll talk about this a little bit later on. So let's now look at the ancient tree and some of the ancient features of it. Starting at the very top of the tree, it has a mature crown. This is an example of an ancient eucalyptus tree in the forest in which I work. This is a tree that's a few hundred years old, and you can see it has a massive accumulation of biomass. There's a lot of bark, leaves, twigs, and branches. There's also a lot of dead branches too at this stage. Canopies often retrench in old age as the tree sheds and the bark drops and the branches start to drop due to decay over time. Canopy soils even develop in the crevices between branches of ancient trees, like here, where organic matter like leaves and twigs accumulate and break down over time. All of these unique features of the mature tree crown mean that there's more diverse places for fungi to live on it and feed on it. Moving down the ancient tree now, ancient trees of course have ancient bark, such as these two ancient oak trees here. Ancient bark is often characterized by an increased surface area of moss and lichens. It often creates these deep fissures and crevices that create little microclimates, and you can see here these two ancient oak barks have quite a lot of colonization of lichen species. The unique bark creates a unique chemical composition. The ancient bark is often higher in pH, and even the water running down the ancient bark is unique and distinct from younger trees. Again, all of these unique characteristics of the ancient bark means that there's more diverse places for fungi to live and feed on it. A really important ancient feature of the ancient tree is the development of heart rot and hollows, such as these pictures here. Heart rot is the decay of the central wood, the non-living wood inside the tree by fungal decomposers, and this can ultimately lead to the development of hollows over decades to centuries, depending on the different tree species. And these are a very important, functionally critical resource for a lot of animals, and we'll talk about this a little bit later on. Ancient old trees also have more alive and dead biomass and a well-developed root system, and of course, abundant organic matter on the soil surface. It's important to note that ancient trees come in all different shapes and sizes. For example, these are some ancient elm trees from Norway, an ancient eucalypt in Australia, a twisted willow tree that's ancient in Sweden, and an ancient oak. Generally speaking, it's not really size that matters, it's age that matters. But an ancient tree is not strictly defined by a specific tree age or a tree size, but rather it's a combination of the ancient features that I just spoke about that indicates that the tree has reached an advanced stage of maturity. For example, some ancient trees can have a large circumference, like this ash tree that you can see with advanced heart rot, or this ancient eucalyptus species that has a circumference of more than 20 meters and is more than 90 meters tall. But of course, you don't have to be big to be ancient and have ancient features. And this lovely ancient elder tree is a good example of that. It has just a very small circumference of 1.5 meters, but you can see here examples of heart rot and hollowing and ancient tree bark on this very small ancient tree. Ancient tree features like the mature crown we just spoke about, ancient bark, hollows and heart rot, select for unique communities of fungi that vary across space and time and with individual tree species. Collectively, we can think of all the communities present on an individual old tree and their many interlinkages as the ancient tree holobiome, which has many important functional roles, which we'll talk about shortly. Essentially, the ancient tree holobiome contains a small universe of life, and I think this will become apparent as we talk through the ancient tree holobiome in the next few slides. So starting in the very top of the tree, in the canopy, and the canopy is quite a challenging environment for any organisms that occur there, including fungi, because of sudden changes in humidity and temperature. But many fungi have adapted to life up in the treetops. For example, fungi can occur on leaves as epiphytes, on leaves as pathogens, or inside leaf tissues as endophytes. They also occur in the canopy soils, and these even include mycorrhizal fungi that form on aerial roots. And the mycorrhizal fungi in the canopy can actually differ quite a lot to the mycorrhizal fungi in the soil on the same tree. So they're very diverse communities. On dead twigs, branches, and leaves, of course, we also have saprotrophs, and this includes a really unique and cool group of fungi, which has recently been referred to as spider-like fungi. This is an example of these spider-like fungi here, or they're also called horsehair fungi. They form aerial root-like filaments that can act as nets, these web-like black structures here. They occur in the canopy of tropical forest, and they can collect fallen leaves and insects, providing little microhabitats in themselves. Moving down the ancient tree now, let's talk about bark and trunk fungi. Fungi can occur as endophytes in the bark, branch, and stem of the ancient tree, and these are highly diverse communities. Recent studies have shown that there can be hundreds of species of endophytes in the sapwood of just a single oak tree from a sample of a tiny amount of sawdust, so they're quite amazing communities. The unique composition of the ancient bark also selects for unique epiphytic communities, such as lichenized fungi. And these are some examples of some great lichenized fungi here that occur on ancient oak species. Many of these species are quite specialized and rare and also threatened and might only occur on ancient trees. These include species like the Lamur Labaria pulmonaria. This species here has poor dispersal and is reliant on ancient tree availability. It also, lichenized fungi on ancient trees also includes those that can be strong dispersers, such as the Scleostomum species that can disperse multiple kilometers, but of course it's limited by the availability of ancient trees. So it's really not enough to have a strong dispersal capacity. You definitely need the habitat availability to disperse too. On the ancient tree trunk, saprotrophs also colonize non-functional sapwood from wombs or latent propagules. We also have canker-forming pathogens. And of course, hollow-making and heart-rotting fungi are abundant in the ancient tree trunk. Some examples here. As I said before, heart-rotting fungi decompose the heartwood to make hollows over decades and centuries. The fungi can colonize the heartwood through wounds on the trunk, roots, or through latently present propagules. Some of these heart-rotting fungi are species-specific. For instance, this fungus, the Fistulina hepatica species, or the beefsteak or tongue fungi as it's also known, occur on ancient trees that have heartwood that many other fungi can't access, such as on oaks and sweet chestnut species of tree. Other heart-rotting fungi, such as malaria species, can be much more generic and occur on lots of different tree species, perhaps because they can enter the tree pathogenically. Under the understory of our ancient tree are unique communities of soil, litter, and root fungi. We've got soil and litter saprotrophs on and in the soil, in the leaf litter, root endophytes that can act as saprotrophs and pathogens, and of course the important mycorrhizal fungi, which are fundamental to the function of the ancient tree. I think you can now understand what I meant by this quote, that an old tree is a small universe. They are really teeming with life, and we've only just spoken about the communities of fungi on the ancient tree. So if we thought about speaking about the other microorganisms on these trees, bacteria, archaea, I think we would need a few more hours to talk about them too. Okay, so as I've just discussed, an ancient tree produces these amazing ancient features that support a diverse array of fungi, including those that are threatened and rare, such as the lichenized fungi. It's important to also note, however, that the ancient tree holobiont has many important functional roles for other kinds of biodiversity other than fungi. For instance, home is where the heart rot is, literally for thousands of species. Globally, over 2,000 species of birds and mammals species nest in hollows, of course, also invertebrates like these. And this includes species that are threatened and endangered. For instance, in Australia, this adorable greater glider, which is an endangered arboreal marsupial, is absolutely dependent on the hollow-bearing trees that occur in the Victorian forest that I work in. Of course, we also have the critically endangered violet click beetle in the UK that's dependent on ancient trees for its survival. Another functional role of the ancient tree holobiont is its ability to provide food for different biodiversity. This often results in a symbiotic relationship where animals and fungi are able to benefit through this relationship, where the animal gets fed and the fungi is able to spread its spore when the animal consumes it. An example of this are marsupials in Australia, such as the long-nosed potteroo and the southern brown bandicoot, two very cute animals as well, which can eat ectomycorrhizal truffles. Often these are in an increased abundance under large ancient trees, and these species help the dispersal of the fungus. These species of animals have also been referred to as ecosystem engineers because while digging for truffles, they actually turn over a large amount of soils, benefiting the soils, promoting nutrient cycling, and also seed germination for even threatened communities of plants. Now, the ancient tree has a distinct life cycle, and fungi interact with the ancient tree throughout all stages of this, from when the tree is alive and well to its death, and of course its eventual collapse. And even in the reincarnation of the tree, if a successful seedling germinates and then hopefully develops into an ancient tree over many centuries. I've purposely coloured these arrows here as rainbows to indicate that there's a lot of stages in between life, death, and collapse. And these often include successional changes in fungal communities over time between these different stages, and they can reflect different decay rates and different colonization strategies of fungi as well. Of course, different fungi colonize the tree at different stages with different tree species as well. So I'm speaking very generally here and throughout this presentation. So we've already covered the small universe that is the ancient tree holobiont in tree life. But what about when the old tree, the ancient tree, dies? When the ancient tree dies, the sapwood becomes dysfunctional. That means it can no longer transport water or nutrients through the tree. The time of death of individual tree species varies quite considerably from hundreds to thousands of years. For example, a species of tree known as the bristlecone pine can remain living for thousands of years, whereas others, like the eucalypt in the forest that I work in, can live for 300, 400 years. So once the tree dies and the sapwood becomes dysfunctional, it becomes a smorgasbord for decay fungi. Decay becomes concentrated in the sapwood. Roots are decaying, heart rot is more advanced. There's a lot of dead wood throughout the whole tree, and including in the canopy. And this can select for the unique communities of saphotrophs occurring on dead branches in the canopy, and also in the wood, bark, and stem of the ancient tree in this dead state. This also leads to an increase in hollow-making fungi and rune colonizers, which are more abundant at this stage, as well as an increase in necrotrophic pathogens, which establish quite easily on the dead ancient tree in the absence of living cells with active defenses that usually repel this kind of fungi. Okay, so eventually, with time, the ancient tree, unfortunately, will lose structural integrity and eventually fall to the ground and undergo advanced decomposition and return to the soil as nutrients. But at this stage, the ancient tree is still functionally important. At this stage, in the collapsed state, the ancient tree's roots are actively decaying above ground. As a result, we see a lot of soil displacement and an increased amount of fungal necromass. There's also a massive amount of nutrients released into the soil with advanced decomposition of the ancient tree. On the trunk of the collapsed tree, we see advanced heart rot and decay of sapwood, and there's a lot of crevices and holes forming on the tree, as well as moss formation and lichen colonization, which can act as nursery sites for seedlings. These factors that characterize the ancient tree in collapse select for lots of unique communities of saprotroph and result in advanced colonization of pretty much all surfaces of saprotrophs and epiphytes. Often in the forest that I work in, the tree is completely green and moss covered at this state. There's really no evidence of wood at all. We also see colonization of the tree by soil fungi, including cord-forming wood rotters. So they're also joining the decomposition party at this stage of the tree. Well, we've just talked about how important the ancient trees are for many reasons. It's really important in this talk that I also acknowledge their reality, and that is that there is a global decline of ancient trees, and this poses a major threat to biodiversity. Threats to ancient trees include logging, wildfire, and climate change. These disturbances can interact and reinforce the effects or likelihood of another, which can also produce compounding effects. For instance, climate change and warming can accelerate the occurrence of wildfires in some ecosystems by drying out vegetation, which ultimately leads to a loss of large ancient trees when they're burnt by wildfire. Similarly, logging can accelerate the occurrence of wildfire by changing vegetation composition to perhaps be more flammable, such as occurs in the mountain ash forests in Victoria and Australia. You can see here just how dense some of the forest is after logging. Of course, when there's an increased amount of wildfire, again, we get a loss of these ancient trees. Together, logging, climate change, and wildfire mean that there is a serious issue when we're thinking about ancient trees, and all of these disturbances together really spell disaster. So these disturbances that I've just spoke about and lots of others that occur in other forests in the world compromise all stages of the ancient tree life cycle, from life to death to collapse, all important as we've just spoke about, but also in the recruitment of ancient trees. For instance, now more than ever, trees are less likely to reach ancient stages. In the forest I work in, there's just less than 3% chance that trees will reach the ancient stages needed to develop hollows to support species like the greater glider that we spoke about earlier, the endangered species, because of the risk of wildfire. Species at most risk of decline when we lose ancient trees are those that have specific relationships with ancient trees and they won't occur anywhere else. And also those species that are dispersal limited, like we spoke about before, the mycorrhizal fungi and also some distinct communities of lichens. An example of this is the lichenized fungal communities that occur on elm species, and they're quite at risk of co-extinction locally because of Dutch elm disease. So it's very clear that time's against us and we need ancient tree features right now. Management practices might be therefore necessary to help fill the gap between the ancient tree development, which can take decades to centuries, and fungi has a key role to play in this. One of these management practices is tree veteranization, and this is described by the process where trees that are of a younger age are damaged to create hollows that are then sometimes inoculated with an appropriate wood-decay fungus. The trees are often damaged using a chainsaw or even a sledgehammer. Sometimes they're even set on fire with small intensity fires. And the aim of this is to speed up the development of ancient tree habitat, like hollows, which can provide for invertebrates and birds and mammals, as we just spoke about. There has been some successful colonizations of inoculated fungi with veteranizations in artificial hollows, but the success is not guaranteed. That's because there may be a lack of conditions that enable the colonization by the fungus. For example, the water content of wood in younger trees is much higher than that of older trees. There's also a lot of ethical considerations that need to be made and best practice guides when we're thinking about these kinds of practices to ensure that no negative consequences result. This includes making sure that we know that the fungus that is inoculated is local and appropriate. And this often means we need to know what heart rot fungi are in a tree in the first place, and we often don't know this in many different trees. There also must be enough resources available to enable long-term research to evaluate the effectiveness of this practice. Of course, while these practices might buy us some time in the long run, there's really no better solution to protecting the ancient tree holobiont than the conservation of ancient trees themselves. Okay, so in summary today, we've talked about how the ancient tree holobiont is indeed a small universe, and that fungi interact with the ancient tree holobiont throughout its life cycle in diverse ways, from the standing live tree to death, to collapse and regeneration, and all the amazing stages between. We've talked about the role of the ancient tree for biodiversity and the threats that they face, how fungi might be manipulated as a tool to accelerate the development of ancient tree features, and the need for the conservation of the ancient tree at all stages, including its recruitment into the future. So I hope you've enjoyed this talk today and learned more about the importance of ancient trees for fungi and fungi for ancient trees. And just before I end, I just want to acknowledge my supervisor, Professor David Lindenmayer, who has advanced my career in many ways and encouraged me to work in these tall, amazing forests, old growth forests in Victoria. And of course, Professor Lynne Boddy, who invited me to talk today and who inspired a lot of the topics that I spoke about, and other authors of the work that I've been talking about today. Thank you very much. So I'll stop.
Thanks, Elle. That was awesome. Beautifully presented, and I'm always in awe of your wonderful drawings. And for me personally, although I've worked on ancient trees for perhaps just about 10 years now, it was really wonderful to see lots of the features in totally different parts of the world. And I think that's one of the great joys of today's webinar, is that we do hear about things that are going on elsewhere in totally different ecosystems with different plant associates, etc., etc. So thank you so much for that. That was super. Okay, we'll now move on to our next speaker, and that is Cherdchai Phosri. And Cherdchai is Dean of Science at Nakhon Phanom University in Thailand. And he's interested in all sorts of things, including plant and rhizosphere microbiome, and development of inocular for forestry practices, etc., etc. And today he's going to tell us about a fungal focus from Northeast Thailand and sustainable production of edible fungi in the Mekong region. So Cherdchai, welcome. It's a joy to have you here. If you'd like to share your screen now.
Okay, one moment. You see my slide properly, Professor Lynn?
Yes, at the moment it's not in the present. That's it. Beautiful.
All right. Okay. So thanks for introduction, Professor Lynn, and good morning, everyone from the north and also from the south. I am very honored to be here with all of you today to present our work on the topic about a fungal focus from the northeast of Thailand and the sustainable production of edible fungi in the Mekong River. So please stay tuned with me for another half an hour. Well, for my talk today, I will showcase you our study from our small lab here. What is the current status of our knowledge of fungi in the northeastern part of Thailand? Where are they found? Where are they distributed? And which are most important species in terms of the economy for the local context here, and how we connect the people, the mushroom or the fungi and the nature together, and how do they, the fungi, help to mitigate the global environment and the economic crisis in the northeastern part of Thailand after the post-pandemic of COVID-19. Well, just give you some briefly idea where my geo position, where is Nakhon Phanom. Probably you're familiar with the capital city of Thailand, Bangkok. Nakhon Phanom is actually a province in the northeastern of Thailand, bordering to Laos and to the east over of the Mekong River. So it is the place where a significant religious belief that is said to be contained of the temple, which are contained of the Buddha's breastbone. So if you get bored with Bangkok and you want to go to see something different from Bangkok or around the center, you can come over here to Nakhon Phanom. It's quite an easy going city. My colleagues, professor from Estonia, he visits us before the COVID. He said this province is very easy, but not lazy. But I mean, it's very chill for relax and also good for retirement. The first group of the fungus focus for here today, I'm gonna give you about what we working on the fungi in the family Salariaceae, which are common, found on wood, leaf, and seeds, and all in soil. Yeah, their food body, and you see from the slide, they are vary in shapes, size, and color. They now have more attract attention from many mycorrhizals around the world because their pharmaceutical and medical properties. The group that we are working with at the moment since 2015 is the Salariaceae associated with termite. In the slide, you see where the Silalia associated with termite distribute around the world in tropic and also subtropical. It's approximately about 32 species were described, and most of them from tropic and the subtropical area, and only 5 species described from Thailand from the year 2015. So this is the example of the Silalia species associated with termite nests from report from Thailand. They are including like Silalia ecumina altissima, Silalia astrodivaricata, Silalia nigipres, Silalia escheridia, and Silalia tankiensis. The pronunciation is a bit difficult for me too, but you can read from the slide easily. Well, we start to examine the diversity and the distribution of the Silalias associated with termite in termite nest or growing on the termite soil since 2015. And this is actually one of the projects for our student who continue conduct her PhD in Liverpool. Yes, so we survey for the sample mainly in the northeastern of Thailand, in the province of like Nakhon Phanom, where we are, and also the neighbor province like Skol Nakhon province, So there are quite a lot of samples that we collect from the dry evergreen forest and mixed dipterocarp forest. And at the end, from this work, we describe about new species, 12 new species from the northeast of Thailand. Their unique characteristics were confirmed by both classical study and molecular analysis, and also with the validation by the expert in this field. For example, Professor Wally and Professor Chu Ming from Taiwan. We named these new species based on their locations and also spatial characters, and also name for famous mycorrhizal as well. Probably Professor Lynne will familiar with some name here, like Margarete. That's a name for Dr. Margaret Wally, for example. And in the 2023, if you want to look more for more detail, the slightly associated with termites, you can follow our publication in biology published in 2023. There's 12 new silent species from Thailand. These works also recognized as the outstanding thesis award in the field of biology and horticulture by the National Research Council of Thailand in the Thailand Research Expo in 2023 for our student. She become very happy up there. What's more interesting about this Silalia associated with the termite is their property. 5 species of our study is exhibit antioxidant and anti-cancer activity against 4 different cell lines. So you can see this is our work. And also, we also identify what the component using the chemist the LCMS technique, and we can identify by comparing the chemical profile with the chemical database. They contain a lot of identified compounds, and there are still at least 40 compounds remain unidentified now. But now our student, she continue her work with the Professor Saga in Liverpool John Moore University. So we hopefully she gonna find more compound from the Silalias associated with termite. Another second group that we will be focused is actually from my PhD thesis many years back ago. You're probably familiar with this fruit body. It looks like a star shape. It's from the genus Asteat in Thailand. In 2004, are described 2 species of Asteat from Thailand, different, distinguished from the European name. Briefly, the Asteat was established by Pursoon in 1801. He described as Asteat hygrometricus, but after work thoroughly and examined the sample from around the world and also in Thailand, we proposed 2 new species of Asteat from Thailand. On the left hand is the Asteat asiaticus, and on the right hand is the Asteat odoratus. These 2 species are very common in Thailand and also very popular for Thai people because both of them are edible. Later on in 2014, we have grant to the remote forest in Thailand, in the northeastern part of Thailand. We survey in the dipterocarp and mixed decidual forest in the remote forest of Thailand in the northeastern, and we discover a new Asteat species. So we name it the Asteas cylindroni, the new species, for the honor of Her Majesty Princess Cylindron on the occasion of the 84th birthday of her father, King Phumthon. So this name under her name as the princess and the king in particular have both actively support the preservation and the study of the rich natural heritage of Thailand. So now in Thailand, we have 3 Asteas species. This photo shows you when it mature, they expand like the flower on the floor. And you can see here, when they mature, the spore turn from the purplish color into brown color. The size of the fruit body quite huge compared to the other 2 Asteas. Later on, there are many continue working with Asteas, and so far using the ITS LSU phylogenetic study, we can resolve the hidden story of the Asteas complex, which person has described one name under Asteas hygrometricus for more than 100 years. So far, at least I would say we have almost about 11 species of Asteas around the world. And I do believe that there are still more Asteas to discover from other regions of the world. For example, as far as I know, from the Indian subcontinent, my colleague, he are working on the new Asteas from India. And also maybe the Asteas from Japan will be reported soon. And also the Asteas from Africa and even South America are awaited for discovery. So this is going to be the end of the Asteas story. No, but actually after we publish a paper on the scientist community, what's next I'm going to tell you is how we continue study with the Asteas and how we bring the Asteas back into local context. Yeah, this is what the project is going on at the moment. We use of mycorrhizal fungi to enhance reforestation with the native Thai species in Thailand. And I thank to Yuki and Elle that she talked about mycorrhizal, so it saved my energy. Don't need to go to talk about mycorrhizal in general. Here, the Asteas come to play the vital role in the project. How? I'm going to show you in the next. Well, if you make a trip to Nakhon Phanom during the rainy season in between from May to June each year, you probably notice that along the road, you will see a variety of the wild edible ectomycorrhizal mushrooms on sale along the road. This is the source of their income and also provide a food security for the community. There are a variety of ectomycorrhizal available for you to eat in the northeastern of Thailand. The top 2 most popular species here in the northeastern of Thailand would be the Asteas odoratus and Amanita species, the yellow one or yellowish orange one. The price at the beginning of the season when they harvest from the forest, it could be from 12 to 15 US dollar per kilo. I have an interview with the local contact. They said they quite worried about the declining of the production of the mushroom they can pick from the forest, especially this season. A lot of Asteas odoratus are harvested from the forest. And we also afraid of the destruction of the forest or the ecosystem. So what we can do to make sure they will have the mushroom available for them in the next seasons. So it's come up with the idea, as far as we know, that the Erythrorchis trees are one of the most important family of the tree in Thailand and in Southeast Asia. They are known to form ectomycorrhizal with hundreds of ectomycorrhizal species, and they rely on the ectomycorrhizal for their nutrient uptakes. And to promote the reforestation by using of the mycorrhizal fungi, this will become our project. So the aim of the project or the framework of the project to reforestation in Thailand, we would like to help mitigating global climate change through enhanced community-based reforestation. Well, actually, if you introduce the tree, I mean the slow growing tree to the community, they might don't see how important, because they want some benefit to return to them if they want to look after the tree. So the idea come together to what the benefit to return to the local community. If we want to introduce the Erythrorchis tree to the community and to protect the environment, we need to concern about how to provide intensive for the community as well, for the benefit that they will get in return. As the astriate is edible, so the idea come to use the astriate with the Erythrorchis. So we team up with the Royal Initiative Project in the province, where they produce 100,000 seedlings per year. And these seedlings are distributed to the local farmer or reforestation program throughout Thailand or in the northeastern part. Well, to work with the astriate, you can cultivate or isolate the mycelium from the fruit body, the young immature fruit body. They produce the very colorful golden yellow on the synthetic medium. But for the nursery, where they produce 100,000 of seedlings, it would not enough for use in the inoculation program. So we turn into the fruit body, the mature fruit body, and we have organized several workshops for the local farmer, local people, how they can prepare their own inoculum using the mature astriate fruit body, and with kitchen machine like blender and clean water. Or even we produce some spore and keep for a longer period in the fridge, sealed in the bag. On the left hand is the spore we scratch from the food body of the astriate, and they can keep it for at least a year. And when you want to use it, you just mix it with the water. Make it like a suspension or slurry, yeah, and you stir well, and then you can apply to the seedlings. This means you have to grow the seedling at once, about a month. So, and when the fruiting body season comes, you can collect the mushroom, the astriate mushroom, and make a slurry inoculation, and you can inoculate your seedlings. Here, show you the astriate mycorrhizal root tip to make sure that your seedlings are infected with the astriate inoculum. It would take you about at least 6 to 8 months to make sure all the seedling has established with the mycorrhizal. This is the character of the astriate root tip. Is quite branching and in brown color. We also check for the seedlings using infection in the seedling using the molecular technique to confirm that our seedlings are infected with the astrea. But the seedling had to be kept in the nursery at least a month or until 1 year before you can transplant it into the field for the reforestation program. This is on the right hand is our seedlings transferred to the very sandy, very drought area in the northeastern part of Thailand, but they still look okay after 5 years. Not at that tall, but yeah, strong. What is more challenging to us is start in before the COVID in 2016, when we got a call from the local farmer, local community in the northeastern part of Thailand. They said they don't want eucalyptus in their community field. So can we help us to reestablish the plantation in their land? So we engaged with the local community. They help us to prepare the land, and they help us to get rid of the stump of the eucalyptus. So this plantation, eucalyptus plantation, about 64 hectares, become our reforestation site. So this is about after transplant, very dry, drought, and very sandy location. 1 year after, they still look okay. At least I will say 60 to 70% still survive in the field. So, but look carefully in this slide. Yes. What happened to the Erythrorchis tree seedlings after transplant? It happened in 2020. We found the first astrea fruiting in our demonstration site. The tree doesn't that tall at all, but they start to produce the astrea mushroom. This one tree from our sampling, they produce about 200 grams per tree. But not the whole plantation produce the astrea mushroom. The price, quite pricey this season, this year, is about 18 to 25 US dollar per kilo. This is the photo of the astrea odoratus from the field that the local farmer they harvest. So at the moment, we have about 10 current demonstration sites around 100 hectares throughout Thailand. The yellow means the project collaborate between different universities with the community, and the green color show the site where the forestry department engaged with the local community. And also the blue color, which means the project conducted by the royal family. So the demonstration sites are widespread from the northeastern to the east and also to the south of Thailand as well. No, we not limit only the astrea, but I would not tell you more about this, but you can follow us in our website or our messenger web page. The other species that we're working with also the Amanita. This photo taken from one of the Facebook from the Thai singer who interested in the reforestation program. So he got the seedlings from us, and then he do plantation in his own land. And then after 3 years, he sent us the photo. He said, very exciting. He can collect the edible Amanita mushroom, and what left over, he can sell the mushroom to the local market as well. So in conclusion, I have shown you our showcase how we connect nature, mushroom, tree, and people together. And I just would like to give you a conclusion that the role of the infinitely small fungi in the nature is infinitely large for us. So they are play a vital role in the ecosystem and also for the humankind. This really means more to us to work with the ectomycorrhizal fungi. Thank you very much, Professor Lynn, and thank you very much, everyone who listened to my talk. I would like to thank you, my team, the younger staff, and also all the institute, all funding to help, to support us to get this work outreach to the local community. Thank you very much.
Maybe everybody could, so thank you very much for that talk, and we'll have some questions in a moment, but perhaps the other panelists could switch their microphones and cameras on, ready for questions. We have quite a few questions, but actually, while they're in my mind, maybe I could ask Yu-Cheng a bit about the cultivated fungi in China. It's amazing the amount of fungi that you cultivate for food and the ways you cultivate them. So I noticed that very often it's in sort of indoor tent structures, but sometimes for some fungi outside. Could you tell us whether more fungi are cultivated in a sort of an inside environment than outside, or whether it's about even, and what are the pros and cons? Why do people sometimes grow under tented structures or indoors and sometimes out of doors?
Okay, because nowadays, for example, in the most dense population area, they don't have much the land, the spaces, and then they have choose this indoor this production. That means they can save the spaces, and also the production is more effective. So they can organize very well. And then because this kind of mushroom industry is very much depends how many population around this area. If you have a big factory produce, for example, one day for 20 tons of the mushrooms, you must near to Beijing and Shanghai, so you can go to market to sell them. But in countryside, if you have a very big factory and the population is not much enough, and then you cannot sell them. So then they have a small factory, or they have very cheap land, and then they use the open land. This model, they can save more money. This is the money-oriented function.
Yes. Yes, of course, of course, of course. So I did wonder, are all of your cultivated fungi grown from inoculum that has been obtained from local sources in China? Because I know problems or potential problems in other parts of the world is that that fungi are perhaps obtained from elsewhere in the world and then cultured in one's own country who obtained these cultures. And then, of course, there's always this slight worry, or perhaps a larger worry, that these fungi that have been obtained from elsewhere in the world may then escape into the natural environment. But I assume that in China, probably all your fungi are locally sourced for inoculum.
Yeah. Sometimes when, particularly in the open cultivation, some cultivate mushroom, the sprouts will go to forest. But normally this kind of mushroom, they do not affect very much the trees. So they are not pathogen because they are saprotrophy. So it is no problem.
Oh yes, I understand that they're saprotrophs, but if, for example, you maybe imported into a country saprotrophs
Orchid.
from another part of the world, then one could imagine that that could be potentially a problem. If it's a species that isn't found in that country, perhaps it might become an invasive species, or even if it's a species found in that country, it could be an invasive genotype or something like that.
Okay, I didn't think that. But normally, if the Chinese products import to the other country, they have well packaged in the plastic bag. So they just from the supermarket to the kitchen, they open the plastic bag and then they cook it. I think in this case, the spores go to the other country. The possibility is not very much.
Yes, yes. No, I understand that from a point of view of consuming the fruit bodies, but I was meaning if somebody wants to grow a fungus in one country, but they import the inoculum from another country, and then they start growing in their own country, that fungus on a very large scale, I wonder if there's a possibility of a danger of that fungus from another country then becoming invasive.
That may be possible because if they have some similar species and you import some competitor, and it may be, but I don't know very much this case.
No, I don't know either, but I think it perhaps is something we need to think of on a worldwide level of about what we are doing about moving fungi around the world. I mean, I don't know the extent it could be a problem yet either, but it's something we need, I think, to think about. Yes. Could I just sneak in one last question? Then I must bring in all the other speakers as well. I was interested about the collecting of Ganoderma spores. And I wondered why you collect them. To start with, I thought, well, is it for a health and safety issue? You didn't want the workers to be breathing in the spores, or is it that you are then using the spores from the Ganoderma for either medicines or for inoculation?
Because many Chinese studies the Ganoderma spores, they have so many substances called the tree santi. I don't know exactly the chemical names. They have polysaccharides and I don't know exactly the other term, but they say they have so many substances. They are very good for health. So,
so they're collecting the spores for
particularly
medicines.
in this kind of Ganoderma spores, they can very much improve the immunity of the human being. So, for example, for the last 5 years, all the Ganoderma companies, they earn so much because the virus time, people just want to getting more how to say 免疫就just I don't know exactly how to say, but people believe when we eat more Ganoderma, then you can reduce any kind of diseases. So
I see, yes.
this Ganoderma is very expensive in China, in fact. Yeah.
Okay, thank you so much. Let me ask some questions of other panelists. And actually, that there are questions about mycorrhizal fungi to all of the other speakers. So maybe I could ask some of those questions on mycorrhizal fungi to start with. So if I pick one that's addressed to Elle first, and the question that was posed was, what are the main species of mycorrhizal fungi on ancient trees? And are they more present in a particular species in the same family of trees? And perhaps I could extend that to asking how different are the fungi, the mycorrhizal fungi on ancient trees than perhaps on younger trees. I don't know if that's known or not.
Yeah, thanks, Lynn. So most of the trees that occur in forests they're mostly ectomycorrhizal relationships. So they're a lot of the trees we think about in forests like pines and oaks and eucalypts form these associations with these ectomycorrhizal fungi around the root tip of the fungus, rather than the other main form of mycorrhizal fungi, which is the arbuscular mycorrhizal fungi that penetrates the root tip. We see quite a big difference in the ectomycorrhizal communities on older trees versus younger trees. Younger trees can often have early successional species of fungi that might colonize immediately after a disturbance. These species might also have different effects on the trees. They might demand more carbon or less carbon. We usually find species that are more carbon demanding in the older successional trees, like some Cortinarius species and Russula species in the forest that I work in specifically. We find that these unique communities. Does that kind of answer the question, Lynne? Perhaps you might have more insights.
Yes, I'm afraid I don't give much insights into mycorrhizal fungi, but I think that was that answered very nicely. Thank you, Elle. And maybe I could address this one to Cherdchai. And it was interesting that you were talking about using mycorrhizal fungi to enhance reforestation in Thailand, to increase dipterocarp trees, and that you were inoculating these locally produced seedlings. So I presume that your inoculum, this sort of follows on from the question I think that I asked Yu-Cheng. I presume that your inoculum is coming again from local sources. I mean, I thought it was wonderful how you described how the local peoples churn up fruit bodies in household blenders to make their inoculum. So I'm presuming that all that inoculum comes locally. Is that right? Chai, I think, has vanished.
Lynne, most of the inoculum we need to spare some like funding or money to buy from the local source. Yeah, but recently we are the border to Laos, there are a lot of the astrid come from Laos in enormous amount as well. Yeah, so but it's the same species anyway, but you still can use that as a spore suspension to inoculate. But mainly we prefer to use our local, to inoculate in the northeastern part of Thailand. Even the Amanita, we also use the local strain here, to make an inoculum. Yes.
Right. So presumably the idea is that these trees will then produce masses of mycorrhizal fruit bodies for harvest for the local economy.
Yes, we're expecting that. It's just like a win-win situation, the local, they wouldn't look after the tree well if they're not think to return their benefit. And the dipterocarp is also slow growing tree. So it would take about 20 to 30 years, you can get the timber from the dipterocarp. Yeah. So it's too long for them. So that's it, like the win-win situation between the university, the government, the royal department, how to enhance the reforestation program. Yes.
Oh, interesting. So I wondered whether there was evidence that over harvesting reduces fruit body production, or whether the decline in fruit bodies for eating is because of decline of the actual amount of forest that there is.
There are some areas they said they cannot harvest more astrid because of the deforestation. But this year is quite surprisingly, I don't sure because the climate situation or climate change, there are enormous of the quantity of astrid to come to the market as well. So I'm not sure they try, the fungi maybe try to survive under the climate scenario or what they think about the climate. So they produce a lot of astrid this year. So at the beginning of the season, it's quite less. The price is very pricey, about 800 baht or almost $20 per kilo. And then afterward, later in the season, because of the enormous of quantity, the price going very low, about $3 per kilo as well.
Ah, right. I mean, this is a question really for all of you, I think. There was a question that came in, which I will phrase slightly differently, but I think it's asking, I think the person who asked the question was thinking that maybe each tree only has a few different species or perhaps one species of mycorrhizal fungus. But I wondered if you could tell us what the diversity is of fungi on the trees of mycorrhizal fungi is on the trees in your area?
Oh, let me get you the idea first. We used to study the mycorrhizal root tip in the Erythrorchis forest. In one single tree, it contains about 69 species of ectomycorrhizal fungi in our Erythrorchis forest. Not only one root tip, not only one species. Yeah, I think it's more diverse. Yeah.
Yeah, so that's amazing, isn't it? And I'm assuming perhaps Elle could answer a bit more. I mean, I imagine the diversity of your trees in Australia, or the diversity of the ectomycorrhizal fungi on your trees in Australia is equally large.
Yeah, I don't have the exact number in my head right now, but I know that the communities are very diverse, and that increases as they, of course, reach that older stage and have all of those amazing little ancient features of an ancient tree. Different communities of trees in our forests as well can have specialized communities of mycorrhizal fungi. I know specifically some Nothofagus trees that we have down south here form very special and unique relationships with some mycorrhizal fungi, and they only form with these particular tree species. So not only are they diverse, but there's also these unique associations that are species specific.
Ah, yes. Yes, thank you for that, Elle. And actually, that segues nicely into a question for Yuki, where I think, Yuki, you mentioned that most mycoheterotrophic plants are quite specific for their fungal partners, but your Erythrorchis altissima is actually very generous and has lots of different fungal partners. So why do you think there might be that big difference? I mean, that's very different from what you said about other mycoheterotrophic plants.
Oh, yes. So that's a difficult question, but very interesting. Actually, we don't have an exact answer, and it's just my hypothesis. But most mycoheterotrophic plants are very tiny. So they extend the root, but only a few centimeter area. They extend the root. So very small area. So maybe inhabit very limited number of fungi is available in such small area. So I think in such a situation, then the specialist is more easier. Maybe specialist can use a fungi that give the more carbon to the plant. It's very effective. But in case of altissima, they are very large size, like a 2 to 10 meter. So they can extend the root. The root cover a large area, like a few meter. So altissima can associate different fungal species at the same time because they are very large. So I think in that situation, the generalist lifestyle is maybe makes sense. Yeah.
That's interesting. So I think I'm right in saying that most orchid seeds are very tiny and they have to associate with a fungal partner very early on. Otherwise, they can't germinate and grow into seedlings. So is that also true for altissima? Does it have to associate very early when a seed is starting to germinate with a fungus? But then from what you said, maybe it can then acquire, the same plant can then acquire fungi, different species of fungi, and have several at the same time. So do they have to acquire their a fungus almost instantly when they start to germinate the seeds of altissima, and then they manage to get more fungi as they grow, perhaps?
Yeah, I think so. So, the first just germinated very early growth stage, the Erythrorchis size only a few millimeter. I think in that case, they are associated with a single mycorrhizal fungi. But after grow and extend, their fungal partner extend more and gain the carbon more and extend more and more.
Right. Okay. So why do they grow up trees? You know, when there are dead or dying trees, why do they grow upwards? You could understand with plants that have chlorophyll and want to get to the light, but do we have any idea as to why it grows upwards?
I think it's an ancient character of the Erythrorchis. It's dependent on the plant phylogeny. The Erythrorchis is included in the subfamily Vanillioideae. It's closely related to vanilla plants. You know, like a vanilla ice cream. It's a vanilla. Vanilla also have a long stem and they climbing the trees. And the Erythrorchis is very close phylogenetically relationship with vanilla groups. So I think they are, how can I say, the plant phylogenetic reason, I think. They are ancient Erythrorchis, maybe it's like a vanilla style. The stem climbing the root and they have leaves, but once they stop the photosynthesis, they lost chlorophyll and leaves and become only stem, climbing stem.
Right.
I think that it depends on the origin of the plant lineage.
Yes, that's interesting. Yes. So perhaps I just ask one more question, but I ought to then move on to ask some questions of other people again. I was very interested that you said that the altissima dies within 2 years, or at least 70% of altissima die within 2 years. So I wondered whether this was to do with their natural life cycle, whether it only tended to be 2 years long, or whether it was to do, you mentioned the fungi in the wood, and maybe it was a nutritional thing. But I wondered whether it was to do with the fact that the fungi which they partner with or parasitize are actually replaced by other fungi. So as we know, during wood decay, early colonizing fungi, and Flebia often is an early, or the species of Flebia we have in Britain are often early colonizing fungi, which will then be replaced later on by more combative fungi that fight them and kill them. And so I wonder whether with altissima it's because their fungal partners have been killed by other fungi which don't partner with them, if that could be why they only live for 2 years usually.
I see. So I think the reason why they died within a short period, because they are very huge body and they need a much carbon other than mycoheterotroph. So I think the fungal partner should be also huge size to support Erythrorchis, you know. So we visited the same forest and monitoring the same individual for 2 or 3 years, and we continue the check the mycorrhizal fungi for 3 years. But during that 3 years, they associate with the same Flebia species. So I think but it depends on the size of the log, but the huge log, they are a big size of Flebia are living inside, and it's continue 2 or 3 years and support the Erythrorchis growth. But once they disappear after succession, maybe Erythrorchis need to move away.
Yes, yes, yes. Very interesting. Yes, very interesting story. Thank you so much for that. Yes, perhaps I'll better move on. There are some questions then for Cherdchai. I think you've partially answered some of these in the chat, but perhaps I could ask them out loud. Are there different xylariales associated with different termite species?
Well, for the record, we found only the termite in subfamily Macroterminis that associates with the pseudoxylaria actually limbs. Yeah, so it's about 6 genera in Thailand. Yeah, for this termite, and we found the Xylaria fruit body when the termite leave the nest and on the soil on top of the termite hill. Yeah, most of the time.
Ah, so they're usually found on the top, and it's when the termites leave the nest. So the
Yeah,
termites are
when the
no longer
yes, not
grooming
there.
the Trichomyces, and so the
Yeah.
xylariaceous fungi
Yeah.
can then get into the nest. Yes, I understand.
Yeah.
I understand that. Yes. One thing I was a little bit confused about, but it may have been me not concentrating properly. On the map you showed at the beginning, with xylariaceous fungi on, I think it was on termites, but I think that I saw that you had arrows to places where I didn't think we found termites, in parts of the world where you didn't find termites. Like I think there might have been an arrow on the map to Alaska or something, but that might have been me getting that wrong.
So, sorry, say again, Lynne.
Sorry, I didn't say that clearly, did I? So I can understand you'd find xylariales in termite mounds around the world.
Yes.
Where we know that there are termites found, so in
mhm.
Africa and where have you. But I thought that on the map that you showed at the beginning, I thought that you had arrows to parts of the world where I didn't think termites were found. Like I think there was an arrow on the map to Alaska or something like that. Or
It's
maybe perhaps I've misunderstood that.
it's normally that's from the paper that report all the termite associated with Xylaria. So I think that's we got like correct the map from the reference. Yeah. So it's the 33 species from tropical and subtropical. Yeah. 33
Yes.
species described.
Okay, I think I might have misunderstood then. And obviously, we'd expect them in subtropical and tropical regions. Yes, I understand. So I also wonder, so you talked a lot about the different chemicals that the xylariaceous fungi make. So I wondered, do different species of xylariales produce very different chemicals than other ones? I mean, I expect some of them, I expect there are some chemicals common to many xylariales, but I wondered about whether there were specific ones associated with specific species.
As far as we know, there are some common chemical in different xylaria as well, and also some specific chemical in some particular species. Yes, so far.
Yes. That's absolutely fascinating. Thank you very much. Perhaps we should move back. I think there are some more questions here I have on ancient trees again. And Yufa Kasawa was asking Elle whether you is interested in regeneration of seedlings on fallen trunks and ancient trees, and you was asking what kind of tree species regenerate on logs in Australia.
Thanks, Lynne. We do find a lot of seedlings generating on decayed, fallen down trees on the forest floor. These are often species that produce seed quite abundantly. So there needs to be a seed source in order for a seedling to germinate. But we find it a lot in the forest that I work in because these are wet forests and rainforests, which means that the ancient tree, when it's fallen down, it's really the perfect habitat. It's moist, it's already rapidly decomposing. So as soon as the seed drops, it really germinates. And when we get that ancient tree falling down, we also get a big gap in the canopy that allows a lot of light to come in. So that enables seedlings to germinate as well. So we've got the nutrients from the ancient tree, we've got the light, it's all happening, and the seeds coming in at the right time as well. So in the forest that I work in, when we have closed canopies and there's not a lot of light, you actually don't get much germination at all until you get this big fall of an ancient tree or an ancient tree branch. And then you can get a new recruitment of seedlings on the tree or even in the soil.
So that leads me to ask a question that's come into my mind. With some of our ancient trees over in Europe, for example, oak. Oak doesn't cope well in a closed canopy situation. It's more of an open growing tree in wood pasture. I was wondering about your ancient trees. Do you get lots of ancient trees in a closed canopy, sort of forest environment then?
As the forest gets older, the density of the ancient trees, it kind of expands quite a lot. So you don't get a lot in close proximity. They often, the forest and the ancient trees, the trees themselves thin out over time. So the density of trees in a younger forest is so much more dense. And as you go on and on and on, they become more open. And of course, then at the old successional stage, the ancient trees, we get about maybe 10 to a hectare. It's not a high density amount of trees because of course they need the space and the nutrients to grow, and the more space, the bigger they will get and expand out. Yeah, so we don't get really, really dense configurations of old trees, but their canopies are so large, they really feel like they do spread out the space and fill out the space quite well.
Yeah. Thank you for that. There are more questions on ancient trees, but
Yes.
I see one here for Yu-Cheng. It's a question about Chinese fungi and growing them. Are there different regulations regarding open space fungi growing and closed space growing of fungi?
I don't think we have special regulation on this open model and the indoor model. I
All right.
don't
So no. So there's not. Is there much regulation at all on growing fungi? Many laws associated with growing fungi. I don't know about that at all.
Oh, there's no such ruler or law. Just there are some organization they will check the quality of the mushroom they produce, but because some mushroom they can concentrate some heavy metals, and then in such case, for example, if there are some factory nearby and there are some pollution, if you cultivate mushroom in this area, and then the mushroom may be not healthy, and then that kind of organization will check the quality, how they are healthy or not healthy, something like that. Yes, I checked term, the Ganoderma spores. They have one substance called ganodermic acid or triterpene, this kind of substance. They are very healthy for human being. So that's why the Chinese people buy this kind of products. Yeah.
Yes, I understand. Thank you. Thank you. Okay, maybe I could come back to Elle now. There are a couple of questions about protection of trees, and I'll combine two questions together, if I might. One question says that, or it was a statement to start with, all trees are a small universe, but have potential to become ancient trees. So all trees and the landscape that they're in should be protected. So maybe you'd like to comment on that in a moment. But a second sort of a follow-up question by someone else, it says, is it possible to protect ancient trees, especially as some indigenous populations prefer to use these trees in this habitat? So perhaps you could comment, Elle, on possibilities for protection of ancient trees in Australia, in your region, I guess, perhaps.
Yeah, so the protection of ancient trees in our region is really just about trying to prevent wildfire occurring and wiping out these trees. In the forests I work in, for example, the old trees just represent just 1% of what's in our forests. So I guess the conservation of trees, you really need to think about how they're distributed within a given area. If there's a lot of them, perhaps there are many different other uses for these trees. I think we need to respect other cultures and need to use trees, but the research is pretty clear in how valuable the ancient trees are for fungi, for biodiversity, producing things like hollows that take hundreds of years to develop and seconds to lose. We really need more ancient. That's the the point is we need more ancient trees in the landscape. But of course, young trees are also very important for fungi, and distinct groups of fungi occur on young trees, middle-aged trees, and ancient trees. But because they're declining and they're at such a risk, I think the focus on the conservation of ancient trees globally is quite warranted. Yeah. Did I answer that? Is the second part?
Yeah, I would agree entirely with that. We really do need to be protecting our ancient trees and getting trees to that ancient stage as well. Actually, what you mentioned there does lead on to another question that has been posed by someone. And they said, you mentioned Dutch elm disease. Are there many fungi causing problems to ancient trees? So in other words, are there many pathogens which are causing problems to ancient trees? Or is it really that wildfires and human felling of trees is the biggest problem to the ancients?
I think, yeah, there's a lot of threats to the ancient trees, and all are pretty much equally important, especially the pathogens that target specific individual trees. Some tree species can survive wildfire, for instance, but maybe they have no chance against a pathogen. We know there's generalized pathogens like the Armillaria species that can make the tree decline over time in terms of its structural integrity. Other major diseases, apart from the Dutch elm, that I can think of at the moment would be ash dieback, is a major pathogen that's affecting a lot of forests and ancient trees, and forests being able to develop the ancient tree stage. Myrtle rust is another big one in Australia that can affect plant species of the Myrtaceae. Wildfire, of course, is a threat to ancient trees, and in some of the forests that I work in, we even see this interaction between forest pathogens and wildfire, where after wildfire we get this big increase in the pathogens, fungal pathogens, and we get a decline in ectomycorrhizal fungi, for example. The pathogens can also benefit in dispersing with wildfire as well. There's some research that shows that their propagals can be aerolized in the smoke after wildfire, so we can get the compounding effect of wildfire pathogenic fungi. So I think, yeah, in summary, they're equally as important, and we need to consider all the threats to the ancient tree and the fungi on them.
Interesting. A question just shot into the chat there, and I'll read it directly. It says, for things like Dutch elm disease, is there any microbiology that can target Dutch elm disease fungi, ash dieback or cowrie dieback in New Zealand? So I'm assuming the question is about biological control there.
Yeah, that does sound like that. I'm not aware of the tools that you might use to control these pathogens. I know that they're expanding into new ranges, and I heard a lot about them recently at a conference that it's such a risk, especially the ash dieback is affecting a lot of new forests that it didn't otherwise. But yeah, I'm not sure, unfortunately, about how to approach targeting the restoration. Yeah.
No, me neither. I don't know the answer to that.
Sorry.
I just thinking about though ash and elm. I might be. I don't know. You'll have to tell me here. I'm assuming that ash was imported into Australia and New Zealand when Europeans colonized. So I'm assuming that ash is non-native. I'm not saying that's any reason to be complacent, because obviously that they're important landscape trees now, and I suppose this same is true of elm. Is that right? That they're non-native,
they're non-native,
but they are important landscape trees now, because they've had
Yeah,
hundreds
and
of
they
years.
occur, of course, in a lot of urban areas as well in Australia. Yeah, but not native.
Yes, and of course, urban trees are very important too. You're absolutely right.
Of course.
So I'm just going to ask one more question of Elle, because I think we've come to the end of the questions which I have, but this will give anybody else an opportunity to shoot in a few more questions, and some things have shot in as I've said that. So I'll just ask you this question, Elle, and then I'll come to these other questions. Actually, really, I think it may be not a question at all. I think it might be a statement from me. I was interested that you mentioned fistulina in your talk as being specific to certain, well, just specific to a very narrow range of trees, and I absolutely agree. In Britain and Europe, we have Fistulina herpatica, which is pretty specific to oak, quercus, and to castania. I think certainly for our European species, the reason that it's specific is that it is co-evolved probably with those trees over millions of years. And it seems that Fistulina can cope with the tannins, the very high content of tannins, the very low pH of the oak heartwood. And most fungi just haven't evolved to cope with that. But I think that's just one reason for specificity. I'm sure there are lots of reasons. Now, let me have a quick look. Okay, I think this one might be actually for you, Elle. So stay on the screen for a moment. So one question says, and I'm reading it directly, they're trying to reforest New Zealand with natives, but the kauri tree, which is native, is also succumbing to a dieback event. And of course, that yes, I do know that's true. In fact, I don't know whether it was last year or perhaps the year before we had someone talking about kauri dieback. I don't know whether you have any comments on that.
I don't, but I'm wondering if climatic changes, I know that it does help facilitate a lot of pathogenic fungi. And so I'm wondering if perhaps we're seeing an increase in New Zealand of that with changing climate. But I'm not aware of that, sorry, that circumstance with the kauri tree. But what you were saying before about the heart rotters and species specific, I think so much work to do. Like in Australia, for instance, we really don't know what a lot of our species specific heart rotters are to facilitate things like, inoculation and veteranization of tree hollows. So I think there's a massive knowledge gap there and something we need to work towards.
I wholeheartedly agree with that. I think that that's true all around the world, and that's certainly true in Britain and the rest of Europe too. We know so little. We've done a bit of work ourselves on oak and beech, and more recently on ash, but there is still so much to do. These trees have really not been looked at with regard to what their fungi are and what they're doing. But yes, thank you very much for that, Elle. And now, actually, this is a very nice comment from John Drury, and it's to everyone. And I think this will be a very nice to get everybody's thoughts on this. Would be a nice close to the session. And the question or the comment is: Yu-Cheng's slides about the use of Ganoderma in arts were fascinating, and I really agree they were. And the question which comes to all panelists, and so please feel free to answer when you want to. Do the panel have other species examples? So I think that's probably of the use of species in art and other areas other than biology and science in general. So would anybody like to say, I don't know this, so this could be, does your country have species of fungi on, I don't know, postage stamps or whatever? Could people answer as to whether their country has specific examples of fungi in the arts? In its widest form.
Sorry.
So Yu-Cheng, the questioner loved your use of Ganoderma in art, and actually is asking the whole panel whether they could tell us more about fungi and art. I don't know whether you've got any more examples from China.
Okay.
And also whether other panelists could give us some examples.
Fungi, when they become dry, they are not beautiful anymore. For example, the other mushroom, they are very beautiful in fresh, but when they become dry, they are ugly looking. Only Ganoderma, they have this very beautiful, this upper surface, even in fresh and dry. So then it is how to say, pokey and woody hard, so they don't decay in when it dry. So that's why we put this Ganoderma is Arctic. In fact, in ancient China, you can see many pictures of Ganoderma 2,000 years ago. So that decoration is not from nowadays, from 2,000 years ago. So we continue to our traditional this kind of artistic model for even they know that we expanded to different parts. So
Yes, that's wonderful. Fungi in ancient art. I wonder, do any of the other panelists, do you have any examples of fungi in art, whether it's ancient, modern, whether it's just, I don't know, anything? Yuki, do you have any examples that you could give for Japan?
Oh yes, I frequently found some Amanita fungi. It's Amanita. It's very like nice shape, and I also have some figure with Amanita with a frog in my desk. Yeah, this kind of figure we can find in Japan. Very nice. It's like a melon. Yeah, it's very nice shape. Yeah.
I think that the Amanita muscaria, the fly agaric, must be probably the most featured fungus in the whole world, in art, children's literature of all sorts, just everywhere. Yes, thank you. Cherdchai, how about you? Have you got any examples for Thailand?
Yeah, there are some stamp collection about the saprophyte many years ago. Since I was a child, I couldn't remember, but I have collected that stamp collection in my stamp book as well, but it's gone somewhere now at the moment. Yes.
That's wonderful. I fear that in Britain we have never had any fungi on stamps, despite trying, but I know that around the world there are lots of fungi on stamps, which is wonderful. How about you, Elle? Have you got any examples?
I can't really think of many. I think Australia is still a bit fungi phobic, and we're just growing, I think, in our interest and appreciation of fungi. But I do know that some Indigenous communities do have ancient cultural features of fungi in some of their paintings. So that's something that's really interesting. Yeah.
Yes, yeah. And I think certainly in Britain, art and fungi is really just beginning to take off in all sorts of different forms, in art, in fungi, in music, and I think there are some forthcoming art exhibitions which involve fungi too. So it is good that science and art are merging. And actually, I'll finish off this session by mentioning something which someone has put in the chat, which I also knew about, and the person says, I've recently purchased a board game produced here in the UK called Mycelia, which has really beautiful illustrations. And that's absolutely true. I have to admit that I had that as a birthday gift, but I haven't played the game yet. So fungi are featuring in all sorts of ways. And I think that's probably a wonderful point to finish. And I would like to thank all of you so much, all four speakers, for giving wonderful presentations. And it's been a great joy and real revelation, actually, to learn about fungi in different parts of the world. And that's what this is all about, and to spread the fungal knowledge. And also thank you, everybody who's posed questions in the chat, and all of you who've come along. So thank you so much for that. It's been a very enlightening day so far. This is only the first session. There's a second session which will be featuring talks from Europe and Africa. And then the final session today will feature talks from the Americas. So I welcome you all to that. I fully understand that those of you who are in the east of the globe probably won't be able to make it live to those sessions, because by then it will be your nighttime, and you will have gone to bed, no doubt. But hopefully you'll be able to catch up when some of the talks, at any rate, feature uploaded onto the British Mycological website. So thank you very much, everybody, and I look forward to seeing you all again soon. Thank you, and bye-bye.
Thank you.
Thank you, everybody. Yeah, have a good
Thanks,
night.
everybody.